Hello friends. Today's topic is pH meter. We all know about pH, but, for revision, just take a look on pH. So, what is the definition of pH? The simple definition of pH is measurement of hydrogen ion concentration. In pH, we measure the acidity or alkalinity of a solution. The equation for calculating pH was proposed in 1909 by Danish biochemist S.N. Peter Lorentz Svensson. pH equation is simply represented as pH equal negative log of H plus ion concentration. We all know that the pH scale usually ranges from 0 to 14. Three types of pH solutions are acidic, basic or alkaline, and neutral. Neutral solutions have the concentration of H3O positive ions equal to the concentration of OH negative ions in pure water. The very strong acids might have a negative pH, while very strong bases might have a pH greater than 14. Remember that not all liquids have a pH value. pH only has meaning in an aqueous solution. Many chemicals, including liquids, do not have pH values. It means if there's no water, there's no pH. For example, there is no pH value for vegetable oil, gasoline, or pure alcohol because it does not contain water. In this slide you can see litmus paper and pH meter instrument. Litmus paper method is easy as compared to pH meter, but it is not accurate method. Handling and storage of litmus paper impact on results. So, litmus papers are not used nowadays. pH meter measurement is quick and accurate. We can check pH of solution very rapidly, and it is time-saving. Portable pH meters are also used instead of regular pH meter. In this slide we know about inventors of the pH meter instrument. A pH meter is a scientific instrument that measures the hydrogen ion activity in water-based solutions, indicating its acidity or basicity expressed as pH. Its working principle is somewhat similar to voltmeter. pH meter first invented by Nobel Prize-winning German chemist Fritz Haber in 1868 to 1934, and his student Zygmunt Clemensiewicz in 1886 to 1963 developed the glass electrode idea in 1909. The modern, electronic pH meter was invented about a quarter century later, in 1934, when American chemist Arnold Beckman (1900 to 2004) figured out how to hook up a glass electrode to an amplifier and voltmeter to make a much more sensitive instrument. He was granted a patent in October 1936. A typical tabletop model of pH meter consists of two main parts. First is pH meter and second is glass electrode. pH meter has on-off switch, display unit, temperature knob, calibration knob, standby mode or millivolt knob. By using on or off switch we can switch on or off the pH meter. Display unit shows readings. The temperature knob is used to set the temperature at which pH is to be checked. Calibrations knobs are used in calibration process to set desired pH of standard solution. When pH meter is not in operation, it is kept at standby mode. For this process, standby knob is used. By rotating standby knob on millivolt side we can check the millivolt also. pH electrodes are constructed from a special composition glass which senses the hydrogen ion concentration. This glass is typically composed of alkali metal ions. The alkali metal ions of the glass and the hydrogen ions in solution undergo an ion exchange reaction, generating a potential difference. In a combination pH probe, the most widely used variety, there are actually two electrodes in one body. One portion is called the measuring pH electrode, the other the reference electrode. The potential generated at the junction site of the measuring portion is due to the free hydrogen ions present in solution. The potential of the reference portion is produced by the internal element in contact with the reference fill solution. This potential is always constant. In simple language, the measuring pH electrode delivers a varying voltage and the reference electrode delivers a constant voltage to the meter. For more information, see next slide carefully and try to identify parts and subpart. Use of pH meter in microbiology. pH meter is used for pH measurement of liquid and solid media. For agar plates pH measurement, the flat bottom electrode is used. pH of the sample is also measured by using pH meter. pH measurement plays critical role in microbiology because it directly impacts on analysis. Operating procedure of pH meter is very simple process, but it is also delicate because of glass electrode. First step is connect the pH meter to power supply. Switch on the button from pH meter. Carefully remove the electrode from storage solution and clean with distilled water and wipe with tissue paper. Carefully dip in the sample container, mix gently and allow to stand. Rotate the knob on pH mode and wait for a few seconds to stabilize reading on display. After taking reading, remove sample and clean the electrode with distilled water. Cleaning of pH meter is important part because we check variety of sample on same pH meter, which may leads to deposition of particles on electrode. pH electrode may carry traces of material to next sample if not cleaned properly. It is delicate work to clean electrode, so do as per following instructions. First, switch off the pH meter and unplug to avoid electric shock hazard. Then carefully remove the electrode from storage solution or sample. Clean the glass electrode with distilled water for 4 to 5 times and finally wipe with tissue paper. Store the electrode in either buffered 4.0 or buffer 7.0 to keep it moistened. For preparation of buffer, refer the next slide. In this slide, pH capsule, pH tablet and ready-to-use pH buffer bottles are shown. Preparation of buffered from capsule or tablet is simple process. In a 100 ml volumetric flask, take initially 50 ml of distilled water and dissolve capsule or tablet. Shake well and finally make up the volume to 100 ml with 50 ml of distilled water. For ready-to-use buffer, just dispense the aliquot or 100 ml in a clean glass bottle. Calibration of pH meter. In this slide we know that how to calibrate pH meter using standard buffer solutions. So, let's talk about basic requirements. We need distilled water is required for electrode cleaning, the tissue paper required for wiping water, and standard buffers for calibration. Remember, the calibration always begin with pH buffer 7.0 solution, as this is the 0 point. Temperature plays important role in calibration. Always set up temperature at 25 degrees Celsius or as per buffer manufacturer recommendation. To calibrate pH meter with buffer 7.0 solution, clean the electrode and dip in buffer 7.0 solution and rotate knob on pH mode, wait for stabilization and note the reading. After that, rotate knob from pH mode to standby mode. Remove the electrode and clean. To calibrate pH meter with buffered 4.0 solution, dip electrode in buffered 4.0 solution and rotate knob on pH mode, wait for stabilization and note the reading and rotate knob on standby mode. Remove the electrode, clean, and finally to calibrate pH meter with buffer 9.0 solution, dip electrode in buffer pH 9.20 solution and rotate knob on pH mode, wait for stabilization and note the reading and rotate knob on standby mode. After completion of calibration, check that observed reading complies to acceptance criteria or not. Please refer the chart for acceptance criteria. If calibration complies, then affix with calibration tag, or if any ambiguity observed, then affix the under maintenance tag. Soak the electrode in a 0.4 molar concentration of HCl hydrochloric acid for 10 minutes, then rinse the electrode with deionized or distilled water. This process removes any organic protein from the glass electrode and the surface of the reference electrode. In case of dehydration, electrode showing sluggish response, the bulb can be rehydrated by immersing the electrode in an ideal storage solution, for example buffer pH 5.00-5.01 solution for 1-2 hours. If this fails, proceed for the electrode rejuvenation.